Upon leaving the endoplasmic reticulum in COPII-coated vesicles, the proteins then move on to the Golgi apparatus for further processing. The Golgi apparatus is possibly my favourite organelle. The reason for this is because it is an organelle that is very much misunderstood in terms of its function. Plus, it was discovered by a pretty interesting guy, Camillo Golgi. The Golgi was discovered by the Italian microscopist Camillo Golgi in 1898. After it was first discovered, the existence of the Golgi was debated for many years as it was thought to be a staining artifact. Its existence was finally confirmed in the 1950s by electron microscopy. However, the microscopy work on the Golgi has led to some misconceptions on how it might work. Okay, so before we get to how the Golgi might work, let's have a look at its structure. The Golgi, when viewed down a microscope side on, looks like a stack of plates. The Golgi has a face close to the endoplasmic reticulum, and this is referred to as the cis face of the Golgi. The face furthest from the endoplasmic reticulum is referred to as the trans face. The plates in the middle of the stack are called the medial Golgi. The role of the Golgi is to complete the processing of proteins passed to it from the ER and then pass those proteins onto lysosomes, the endosomes and the plasma membrane. Hence, it's a processing and sorting organelle. Now, if you think about it, the Golgi could work in one of two ways. That is, the proteins that are trafficked to it could be taken to the machinery that processes the proteins, or the machinery that does the processing could be brought to the proteins. And this thinking has led to two different views of how the Golgi may work. The first model is the Vesicle Transport Model. In this model, the proteins are bundled up and moved from one Golgi compartment to the next in vesicles. That is, the proteins move to the machinery. In the second model, the Cisternal Maturation Model, the proteins stay in place in the Golgi and the machinery comes to them, again via vesicles. Personally, I prefer the Cisternal Maturation Model as there is a growing body of evidence that supports this approach. First, as we've already seen, as the vesicles leave the endoplasmic reticulum, they fuse and form vesicular tubular clusters, and these clusters look very much like the cis face of the Golgi. Another piece of evidence that supports the Cisternal Maturation Model is the movement of vesicles between the Golgi stacks, as these vesicles are too small to carry some of the very large proteins that are processed by the Golgi. Hence my liking for the Cisternal Maturation Model. So, in the Cisternal Maturation Model, the vesicles from the ER form the new cis face of the Golgi. And this new cis face gains the enzymes it needs by COP1 vesicles trafficking back from the old cis face. The old cis face gets the enzymes it needs to become the new medial Golgi from the old medial Golgi, again using COP1 vesicles. The old medial Golgi becomes the new trans-Golgi by getting enzymes from the old trans face, and the old trans face then forms vesicles that go on to form the trans-Golgi network and the endosomal system. As the vesicles leave the old trans face, there are three options for their transport. One, the direct non-specific transport pathway to the cell surface, also called the constitutive secretory pathway. Two, the specific transport pathway to the cell surface via secretory vesicles, also called the regulated secretory pathway. And three, they can go to the late endosomes. So what I'm saying is that instead of viewing the Golgi as a static stack of plates, it should be viewed as a dynamic and ever-changing series of vesicle fusion and trafficking events.